Hello, everybody. Welcome back to another wonderful lecture, the second half of a two-part lecture today. This video is going to be looking at the replication of DNA. So let's go. And this question for this section is, how does DNA replicate? Pretty straightforward. I like it. Three new vocabulary to focus on as we're going through this: semi-conservative replication, semi, partly conservative, keeping, replication, to copy. Okay, DNA polymerase, ACE means that it's an enzyme, and Okizaki fragments. The name gives it away. It was discovered by a Japanese gentleman. The word template is our review vocabulary, and this is a molecule of DNA that is a pattern for the synthesis or creation of a new DNA molecule. The word template generically in English is just a copy or a pattern that can be reused over and over. Magazines and your papers, newspapers used to have printing templates where you would just fill in the spaces and then you would print it over and over and over again. So this is the same kind of idea, only with the DNA in your cells. So during semi-conservative replication, your parental strands of DNA are separated apart by a protein called DNA helicase. Okay, and so you have a DNA molecule, and again in the middle, you have your nitrogenous bases. They're hydrogen bonded, and there's a protein that comes along and unwinds the DNA and then pushes it apart. Semi-conservative replication has three main stages involving bases, pairing, and joining. And so the first is unwinding, which is done again by DNA helicase. ACE is an enzyme that splits something, okay, and so it unwinds and separates the DNA, also prevents it from tangling. Then a single stranded binding pattern begins, right? And so to keep the DNA sort of strands separated, you have to have some other proteins that come and stabilize the structure. Okay, then DNA primase adds a short segment of RNA, which is another molecule we'll talk about in the next lecture. It's ribonucleic acid, and it's called an RNA primer on the end of the DNA strand. And so this is priming. The word priming most commonly used to be used when you're talking about like an engine. You would prime the engine, you'd pump it with a little bit of fuel, and then you'd push the piston down, and it would then ignite. You know, priming means that you're going to set something up to happen. And so base pairing then occurs as the enzyme DNA polymerase adds the appropriate nucleotides, moving from the 3 to the 5 end. I'm sorry, the 5 to the 3 end. No, 3 to 5. It's just not saying it there. Oh dear. There's been a deletion, a mutation. Ooh, Mufasa. I'll fix it in the next go. Anyway, we'll continue. The leading strand is built continuously, and the lagging strand is built by adding smaller pieces together called Okizaki fragments, right? So in one direction, the 3 to 5, it's just adding one base at a time with DNA polymerase. In the opposite direction, you have 3 base pairs, fragments or chunks, that are then attached and then sealed in. So DNA replication occurs in opposite directions at the same time. Then finally, the joining is the last step, where the DNA polymerase removes the RNA primers and fills in the place with the appropriate DNA nucleotide. And when the primer has been removed or replaced, the DNA ligase, which is another enzyme, links the two molecules or sections together. Okay, so you can see there's already a bunch of proteins involved in this. This is a really complicated process. You've got DNA helicase, you've got DNA polymerase, you've got DNA ligase, all working together to allow this to happen. And then you also have primers, RNA primers, and other spacing proteins that hold things together. And so that's really interesting. Now, it turns out that prokaryotes and eukaryotes do this a little bit differently. Is that surprising? I guess not, because eukaryotes are much more complicated than prokaryotes. Eukaryotes carry out DNA replication at multiple bubbles at the same time. So multiple areas unwind and begin to replicate simultaneously. The reason for this is if you tried to replicate your DNA of say a human from one end to the other, just starting at one end and replicating all the way through, the cell would die before it finished. It would just take too long. So by having multiple replication bubbles form at the same time, you can copy it much more quickly, and then the bubbles eventually merge with each other. Now, prokaryotes, in contrast, have a relatively small amount of DNA, hardly any to speak of when compared to, say, a human or even a fruit fly's amount of DNA. And so because it's one big loop, it will just start at one replication fork and unzip all the way around and create two new copies. And each copy has one old strand and then one copied new strand. And then on the bottom, I guess you'd have the copied new strand and the old strand. Hence the idea of semi-conservation. So back to that question, how does DNA replicate? Well, it replicates by unwinding, unzipping, having some binding occur, some DNA polymerase action, the attachment of DNA helicase to zip things together, and it happens in a way that is semi-conservative, an old strand and a new strand, and a new strand and an old strand. If you have questions, of course, ask me. I love questions, but otherwise, thank you so much for listening.